Okay, so welcome everybody to this KBase webinar about fair data in KBase and specifically the GROW database. My name is Zach Crockett. I'm part of the KBase outreach team, and presenting with me is Mikayla Borton at Colorado State as part of the GROW program. And so I'm going to introduce some of the fair data principles and talk about how to use KBase for data reuse and citing data, and then Kayla will talk about her use case with GROW. So to start, I'll talk about the fair data principles and then how those connect within KBase to other research products, and then discuss custom databases in KBase, and then we'll go through a demo of the GROW database. So if you're not familiar with the FAIR data principles, they were laid out in 2016. And FAIR stands for findable, accessible, interoperable, and reusable. And so these concepts are immediately obvious, the value to a person for these data to be findable, accessible, interoperable, and reusable. But they're also important for making data products machine actionable so that computers and programs can more thoroughly reuse data. So in these FAIR principles, the first part, the findable part, is ensures that the data and metadata are findable globally. This usually is most important in getting persistent identifiers for data products. In KBase, we can get DOIs for narratives, and those DOIs resolve to the narratives as a data set. And then accessible metadata means that accessible data and metadata means that the data can be retrieved in a standard way. So in the case of KBase, these are accessible on the web. KBase also uses standardized formats for genomics data, such as FASTQ files for reads and FASTA files for assemblies. And our protocol is open and free and universal, so all of these tools can be reproduced. Theoretically, if you wanted to, you could reproduce all of KBase because it's open source. And then the data and metadata should be interoperable, so they should be able to be used across research projects and institutions and computer systems. And so these metadata use vocabularies that follow FAIR principles, such as the environmental ontologies for environmental metadata and annotation ontologies like the KEG ontology for annotations. And then finally, the R for reusable means that these data and metadata have the correct attributes so that they can be understood in context and can be reused clearly with other data sets. And so for why to make data more fair, the obvious answer is that science can build upon other science if the data are more interoperable and reusable. But as I mentioned before, it's also about making it more machine actionable. And so one of the effects of this is that if data and metadata are machine actionable, it's easier to track credit. And so if you produce data and someone reuses it, then well-structured, fair metadata allows those services to track reuse and attribute it back to you, the original creator of the data. And so the concept of reusing data and making data more fair seems at face value like something that should be obvious that everyone should want to do or be able to do. But in reality, there's a lot of concerns and barriers to making data fair and to making it more reusable. And some of these are described here in this image from Gomez et al. And so in KBase, we try to alleviate these concerns as much as possible. And as some of these may be obvious to a platform like KBase, such as lack of time, which a platform will save time in save time for installing software and for managing large data sets. And some of the other things KBase does not directly address, but can assist the wider community, such as lack of incentives, where we can provide you with citations for your data and give you more incentive to actually make it usable when you get some tangible measurement that you are getting credit. And so all of this kind of hinges around persistent identifiers, which are unique permanent identifiers that can track anything. So in general, in the scientific publishing, we think of DOIs, but there are persistent identifiers for things such as websites. ORCID's IDs are a persistent identifier for researchers. Um, research organization registry or ROR numbers are a persistent identifier for institutions. And so all of these, if we can assign them to data products and people and organizations, we can use those to connect all these different products and people. And and this allows us to expand the relation between these products. So in general usage, you will probably see a person that publishes a publication, and that's where the scientific record exists, is in publications. And those publications may describe a software or a protocol or a data set or a sample. And a lot of times the only persistent identifier is publication. But if we work with people and organizations to expand what persistent identifiers are used and where, we can have a more rich mapping, this citation network that describes the different ways that different persistent identifiers and the things they represent interact with each other. And so instead of having one DOI for a publication, we have DOIs for the publications, sometimes the data management plan, the protocols, sometimes software, and we have persistent identifiers such as RAID, which is something developed in Australia to group an entire research project under one umbrella. And we have IGSNs, which identify samples, and ORCIDs and RORs, which I mentioned before, which connect the people and the organizations to the products they create. And so now I'm going to talk about how that actually works in KBase. And so part of the KBase infrastructure is that it centers around the narrative and apps, which are in this orange section in the middle. And so all of our apps have citations for the original tools that the original tool publication that was used to describe the apps. And so a researcher can bring in their own data or public data from KBase, and we are expanding our tracking of the credit metadata for that to ensure that it gets tracked back to the original source, such as NCBI or NMDC. And then KBase users do some sort of transformation on the narrative, and they can publish that narrative as a fair narrative, which is a static narrative with a DOI. And from that, we can track reuse of that fair narrative. And if a second researcher discovers that narrative and finds some use out of it, they can copy it import their own data, and then produce their own fair narrative. And we can track the connection between those two things to ensure that everyone involved in the process gets credit for what they've done. And so in an actual use case, we have the Genome Resolved Open Watersheds project, um, which has almost all of the identifiers I mentioned before. We have a funding DOI for a CSP proposal. There are IGSN numbers for the samples. There is a protocol for the metagenome extraction, a KBase fair narrative, which I will show shortly, a software DOI for DRAM, which is just um, sorry, I'm blanking on the
Distilled and refined annotation
distilled
of
and
a
refined. Yes,
Yeah.
distilled and refined annotations, and the bioRxiv publication, which is in revisions. And so these are displays of how these connections would actually be seen. So the fair narrative um, has citations for all the data and tools that are used. The publication, which is connected to this fair narrative, has ORCID IDs for the authors. The fair narrative is findable at this DOI and is registered in DataCite, and the work can be found on Mikayla's ORCID page. Okay, and so now I'm going to talk about some of the databases in KBase and how they're set up. So we have this fairly recent section of the dashboard that you may have noticed within the last couple years, the last year or two. We have these collections, which are large data sets that have been put together in partnership with groups such as GROW. And you can find genomes, mags, sample information, and a lot of other relevant data from these projects that you can use in conjunction with your own data for analysis. Um, and so these are a specific thing that is available, but there are also ways to produce your own, not necessarily a collection like that, but you can group um, data in a way that makes it more usable for yourself or for other people. Um, I have included screenshots here from another another collection produced by a user, the produced water DNA database, which has a large collection of published data sets from produced water environments, which are environments that have been contaminated from fossil fuel production. And so in this middle screenshot from their parent narrative, you can see the large selection of samples that are in this data. So there are, for example, there are 144 metagenomes and 2,358 mags. So something like this, if you wanted to share it with other people, would be pretty unwieldy. It's definitely doable and not necessarily hard, but it's time consuming um, and frankly a waste of time and storage space if a bunch of people are working on the same data. And so in a system like KBase, you can create your own narratives and then make these narratives public. And then everyone who is in your same subdiscipline can access them and get to the data that you have produced or you have collected as part of other similar published data sets. So if you're interested in doing something like this, we are excited that several people have shown interest in this. The produced water database that I mentioned before, you can find all their narratives if you search in the public narratives, if you search the username PWDNA. That one's still a work in progress and is not completely finished. And we're starting on working with a group producing a similar concept for fermented foods. So if you have your own type of data that you would like to collect, please reach out to us. We'd love to work with you to help put one together and use some of the lessons learned from the other examples to help you make it more usable for secondary users. Okay, and so I'm going to quickly go through the GROW data that's available on KBase before I turn it over to Mikayla. So if you have a KBase account, you can join this organization. This organization, the GROW orgs in KBase. I'll put the link in the chat. And so this has several narratives with different types of data. The one that I would start with is the samples narrative, which is the kind of the central parent narrative that connects everything. And so in this one, you can see the high-level summary of what is involved in this project and a map of all the sample locations and links to the other main narratives. And we've also got the sample set, which is a KBase object type for sample metadata. And this one is okay, yeah, so this one it has the sample metadata for each of the samples that are in this data set. It includes the controlled vocabulary terms like latitude and longitude to help you sort these samples for specific areas, as well as the common names and the collection information and curation information. So who is responsible for these samples and the associated data. And so if you take a look at one of the child narratives, you can see that the sample set is also in this one for context. And in this one, we have 2093 mags and annotations for those mags. And so those are linked to the samples. So if you do something with any of these mags, it can still be connected to the original sample so that any subsequent analysis you do is connected to that original sample collection location. And this other main narrative is the metabolic models, which is, again, mostly the same idea, but you can find this different data type in this narrative. And finally, I'll show the collection, which I mentioned before, which is in the collection tab. If you go to the beta collections, you'll be able to find this narrative, and you can see this in a slightly different presentation. If you have any questions about using any of these, I'm available in the chat and also always available on help board or engage. And so with that, I'm going to turn it over to Mikayla to talk about the GROW project a little bit more.
Okay, well, hello everyone. Thanks for having me today. As Zach said, my name is Mikayla Borton, and I'll be talking to you today about my work at Colorado State to inventory river microbiomes across North America. So rivers, I can't see. Okay. So rivers are an important connection between terrestrial and marine ecosystems. And so what this figure is showing you is how this connection might impact microbial communities. And so rivers flow from smaller order streams or headwaters down a flow path and interact with the land over this flow path. And so you can imagine that these interactions with different land uses might have impacts on the microbial communities and their function. And so we know that microorganisms play critical roles in these systems. And so this is things like greenhouse gas production, nutrient cycling, and processing contaminants. But despite these microbial communities' importance, prior to GROW, microbial community and viral studies from rivers were scarce. And so this was especially true from a genomic perspective and from a biogeography perspective. And so what this graph is showing you is over the last few decades, the number of biogeography studies in different ecosystem types. And so inland water bodies is this light blue color. And so that's where rivers would fall. And we had very, relative to their soil and ocean counterparts, we had very little information in these systems, and specifically genome information. And so to fill this need or this gap in what we know about river microbiomes, we catalyzed a community effort to sequence global rivers, and this is called GROW. So the Genome Resolved Open Watershed effort, and really it's a database of microbial genomes. And so the map that I'm showing you is all the sampling locations of GROW. And so each point on the map is a sampling location, and the size of the point is the number of samples within that location. And then the color denotes what project specifically within GROW it came from. And I'll talk about a little bit about how GROW was structured to begin with and what those colors mean in a little bit. But we did end up getting about 250 points from rivers, from 250 rivers worldwide, and this included most of the largest rivers across the globe. And so this effort, you can imagine this is a global effort, right? This couldn't have been done by a single lab alone. And so really this was a community effort to sequence global rivers. And this was not, none of the people on this picture are united by a single grant to study rivers and to actually go do this sampling. And so really what catalyzed the GROW effort was a joint Genome Institute supported community sequencing project. So at the JGI, we wrote a CSP. Kelly Wrighton at Colorado State led that CSP. And we brought together people who already work in rivers and who were collecting samples to kind of sequence global rivers. And so all of the collection was done by individual labs shown here, or they were done through crowdsourcing to other scientists already working in rivers. And the crowdsourcing bit, that was really led by WONDERS, or the Worldwide Hydrobiogeochemistry Observation Network for Dynamic River Systems. And that's led by James Segan and Amy Goldman at Pacific Northwest National Lab. And so today, when I start talking about some of the science, I won't be talking about all of these points because most of them, some of them are still being sequenced. So we get, new samples daily coming back from the JGI. But I will be focusing on the WONDERS samples. So those coming from Pacific Northwest National Lab, and then a subset of samples from Byron Crump. And they are shown here. Okay, so WONDERS, I do want to touch based on how these specific samples were collected. So the WONDERS effort out of Pacific Northwest National Lab sampled all of these locations. You can see the locations on the map. Each point is a sampling location, and they actually sent out free stream sampling kits for sediment and surface water to collect samples with minimal constraints. And so these folks that we sent samples to, or sampling kits to, were able to collect samples from whatever river they were working in, or what have you. And so samples were sent back to EMSL, or the Environmental Molecular Sciences Laboratory, for metabolomic analysis. So this is FTICR. Basic geochemistry was done at PNL. And then all of the microbiome samples were sent to Colorado State University, and we processed those here, and those were sent to JGI. And so, as I said, GROW is really just an ever expanding resource. And so the data that I'm going to be talking to you in the next couple of slides is the US surface water focused story. If you remember that map, there are many, many samples beyond that, and we continuously get more samples in. There are also samples, I'll be talking about surface water, there are also sediment samples in GROW, as well as biofilm samples, and those are being uploaded into KBase, which again, I'll talk about in a little bit, but I want to stress surface water, United States. So these are all the data types that we have within GROW, and so metagenomics, and so that did come from JGI, and so what this really tells us is who is there and what is their metabolic potential. We also pulled geospatial information to really put those microbiomes in context of their ecosystem. We had FTICR data, so organic carbon chemistry, geochemistry, which would tell us things like the electron acceptors available to the microbial community. And then we also had metatranscriptomics to think about transcription and expression of what expression of the microbial communities, what functions were they actually expressing. And so today I'll be focusing on the United States. And so this map is showing you the watersheds within the United States. And so rivers are organized into watersheds. And so you can think of this as like a neighborhood within these river systems. And so in the United States, there are 19. And so on this map, they're labeled 1 through 19, and then each watershed is colored. And so we use these neighborhoods to guide our sampling and frame our sampling effort, as well as frame what questions we could ask based on the samples that we had. And so here I'm showing you all the GROW samples. And so each point is colored by the type of microbiome data we had. So for many, we had paired metagenomics or meta-G and metatranscriptomics or meta-T. But for some, we did have only meta-G, and some we only had meta-T. And so if we look across all of these watersheds, you can see that we had samples in most watersheds. And for many of them, we had quite a few samples. And for the regions where you see little to no sampling, so like watershed 9, those are places that we're actively looking to engage with researchers working there so we can get new samples. So from all of these samples, we recovered over 2,000 unique metagenome assembled genomes, or MAGs. And we did this using 3 different assembly approaches. And these genomes were made up of 27 phyla. And the majority of those genomes came from the proteobacteria, the actinobacteria, and the Bacteroidota. And this is really consistent. Again, I'll stress that before we didn't have a ton of biogeography sampling of genomes from rivers. And so we were interested to see if we did have a lot of 16S data. And so we saw a lot of overlap with the lineages that we saw in our MAG data set with previous 16S rRNA studies from river systems. We were then able to take this genome data set and map the microbial expression back to the database to understand the microbial activity in these ecosystems. And so we found that about 25% of the genomes in GROWdb were active in at least 10% of the metatranscriptomes we had across the United States. And so with each of these microbial data types across rivers, we were able to then start evaluating the spatial and geochemical features that influence the river microbiomes. So if you'll remember, we were able to, for each point, one of the reasons I think GROW is really cool is it does have a lot of contextual information in terms of where the samples were collected from. And so we were able to take a point where the sample was collected from, get the latitude and longitude of that, and then pull different types of data, geospatial information, to ask questions about the microbial community in relation to things like land use. So percent forest, percent cropland, percent urban of the watershed that the sample came from. Do we see correlations between land use and the microbial community? Same thing, we were able to pull things like river characteristics, things like stream order, length, mean annual flow, and how do these relate to the microbial community. And so to do this, to actually pull all of this geospatial information, we teamed up with the geospatial centroid at Colorado State University, Matt Ross's lab, to collate watershed characteristics and land use for every site, as well as geochemistry information where it was available. And so these are some of the things that we were interested in. And so our first question at a bulk level was, do microbial communities relate to these features? And one feature that we were particularly interested in was stream order. And so stream order gives us a proxy for the size of the river. So a first order stream is small. So you can think of this as something like Como Creek. And then you can see this picture is a very small stream versus something very large like the Mississippi River. And this would be stream order 10. And so we had a lot of representative from each type or each stream order type. And you can see those here. These are the samples we had. And so we hypothesized that this factor in particular would explain a large proportion of the microbial community variation and structuring, because it's directly related to the amount of inputs on that microbial community. So you can imagine in a small order stream, land use has bigger impacts, and in a larger order stream, land use might have smaller impacts. And so we hypothesized that this might be a key feature that structures the microbial community. And so we found that stream order did in fact structure microbial communities across rivers. And so this graph that I'm showing you is in an MDS. And so each point on this graph is a microbial community, and they're colored by stream order. So you can see from going from left to right that this graph is going from dark blue to dark red, and that smaller order stream microbial communities cluster together, and larger order streams microbial communities cluster together. And so this was a really cool finding that we were able to ask this question based on having all of these biogeography disparate samples. We also wanted to know at a bulk level what other factors we surveyed were significantly related to microbial community structure. And so you're looking at metagenomics on the left and metatranscriptomics on the right. And so we were interested to see which of these features were significantly related to the meta-G, so the metabolic potential, or the metatranscriptomics, or the expression. And so in both factors that were important in both types of data were stream order, sulfate concentrations, chloride concentrations, and temperature. And those are highlighted here on the left. We were really interested to know about the land use, though, because we really thought that this might be something that is directly related to microbial community composition and function. And so we, at a bulk level, in terms of the meta-T and the meta-G, at that level, we did not see, that those had significant impacts. But again, that's the whole microbial community. When we started thinking about specific functions that might be significant relative to land use, we did find that antibiotic resistance genes and antibiotics efflux pumps were significantly increased with wastewater treatment plants. So if a wastewater treatment plant was present, we did see significant increases of these types of genes, and specifically the expression of those genes. We also wanted to know who the core and active microbial community members were across rivers, and then what were their functions. And so this seems like a basic question, but because we didn't have the genomic information previously, we were able to ask this question at a new sort of resolution that we didn't have before. And so across all 62 metatranscriptomes, we found that 6 genera were active in every sample. And some of them were the canonical lineages that you consistently see in river microbiomes. So organisms like Polynucleobacter and Methylopomalis. Many of the core active genera were also the most dominant genera across surface water. And so this includes Methylopomalis, Polynucleobacter, and members of the Burkholderia. And so we next wanted to know, how do expressed functions change across US rivers? And so to do this, we annotated all of the microbial genomes using DRAM. And so DRAM is the distilled and refined annotation and metabolism tool. It not only annotates your genomes, but it summarizes those annotations into things like functional traits to allow quick analysis of the metabolic potential of thousands of microbial genomes. And so, as Zach alluded to, this is available as a standalone tool, but it is also available in KBase for users who want to use it. And so we found that aerobic and light-driven energy metabolisms, as well as methylotrophy, were shared across our core and dominant members. And so you can see the different metabolisms here, but also these top 4 members, these are the most dominant, and we did see that they all encode aerobic and light-driven energy metabolisms. If we focused on the 3 members that were dominant in at least 3 samples, we could see that light-driven energy metabolism was a key feature of these dominant members. And if you look at this map, we could also see that these members really had a pattern. So Methylopomalis was primarily more active on the coasts. Volcanococcus specifically was kind of in the southeast. And Polynucleobacter was in a kind of a middle strip of the United States. And so what was cool is that each of these members actually employed different mechanisms for light-driven energy metabolism. So Methylopomalis used proteorhodopsins, Polynucleobacter had proteorhodopsins and anoxic photosystems, while Volcanococcus had oxygenic photosystems. And so we can start to understand how these different microorganisms make a living in these systems at different parts of the United States, depending on where we were at. And then if we tied it back to some of that geospatial information, we found that certain members, like the Volcanococcus, were correlated to temperature. So higher temperature selects for Volcanococcus. And so that's shown by this density plot here. So we talked a lot about energy metabolism, so aerobic and light-driven metabolism, but what about the carbon? And so a hypothesis in river systems is the river continuum concept. And so it's often thought that inputs matter less. And so you might see larger carbon inputs being utilized by the microbial community in smaller order streams relative to larger order streams. And so we had already seen from our data that stream order was definitely impacted in terms of the bulk microbial community. But we also wanted to know at the functional level within the microbial community, do we see differences there? And so we used DRAM to classify all the carbon genes into different types of carbon. And so we classified them as polymers, sugars, aromatics, methane type metabolism, C1 compounds, and then short chain fatty acids. And then ask the question, do we see these change across stream orders? And what we found was that they do in fact change. And so you can see along this graph, stream order is along the bottom. And so you can see that polymers are used in smaller order streams and less so in larger order streams. But then we see an increase in use of smaller compounds like C1 compounds, and that's this light purple color. And so one thing I really wanted to highlight in this talk, but also as a project for Grow in general, is that we really wanted to make this resource community accessible, not only because we wanted to have, practice good standards and have fair data, but also this is a community effort. And so it was really imperative that all of the people on our team and across the globe could actually access this data. And so we did this in a number of ways. And I'm going to talk about each of them. So the first way we did this was through the National Microbiome Data Collaborative. So GROW is a pilot project for them. And so if you'll remember, GROW has many types of data. So it has FTICR data. It has microbiome data that's coming from JGI. It has a lot of data in geospatial data that's coming from ESS Dive. And so NMDC is a place that links all of those data types together so folks can go utilize each of those different parts if that's what they're after. We also generated a GUI interface for interacting with the data types. And so this is really meant to allow folks, scientists, regardless of scientific background, to interact with the data. So you don't need to necessarily be a microbiome data scientist to utilize this data. And if you're interested in specific samples, you can go see the types of traits that are available in your samples. You can also correlate if you're really interested in something like methanotrophy. Here you can correlate methanotrophy across all of the growth data sets to all of our geospatial information. So does methanotrophy correlate to temperature or pH or whatever it is? And so this GUI interface has really made it possible for a distilled version of the data to be usable to many people. And then the last one that I want to highlight is KBase. And so the KBase team has we've worked with a lot of folks at KBase to make this resource available through the KBase web interface. And so that's what Zach showed you in terms of the narrative. The surface water data that I talked about is available in KBase, the whole data set. And then there are other data sets that are coming online. So there is a Congo data set. There's one from Honduras that's coming online, and we're continually adding data to these narratives and making it publicly available so that other folks can use it. And one thing that I really want to highlight about KBase is that folks can use this data and interact with this data regardless of the computational infrastructure that their lab may have. And so it's free to use and publicly available, and people can do whatever they want with the data and process it however they want in this interface using the many, many tools that are available in KBase. And then in terms of fair data and reusing the data, working with KBase has enabled education and community-based research publications. And so one example of this is we were able to use the GROW database in KBase for a workshop at ISME in Cape Town, South Africa last year. And so this this resulted in a participant-led publication, actually, to a workshop report and a microbial resource announcement. And so from that, folks across the globe were able to use GROW data as well as learn to do different types of microbiome analysis actually on GROW data. And so ISME hosted 25 scholars from African nations, and that is the in-person participants. And so we taught an in-person workshop using data from the Congo, so specific to the African region, and went through the different steps of processing this data. We also had a virtual component where we were able to reach around over 200 people from every continent. And so that was really exciting to be able to utilize the GROW data in this way. And because it was set up in a way that was fair and reusable, we were able to do this. We, as a up and coming way that we're doing a similar approach is at ISME LAT. So ISME hosts regional meetings. This one is in Mexico, and we're teaching a GROW workshop utilizing data from Honduras. So again, using GROW data that is from the region where we're teaching to host this workshop at ISME LAT, and that is going to be taught in Spanish, which we're really excited about. So many of these resources we're making available in that way. Other ways that this GROW data has been reused because of the way that the project was set up to be fair is we were able to lead a crowdsourced publication effort with the FTICR data specifically. And that was in my postdoc with James Stegan and Amy Goldman at Pacific Northwest National Lab. And we were able to put together a series of crowdsourced publications. So many of the people who collected the samples for that Wonders effort actually wanted to tell crowdsourced publications. So we had a series of 6 different questions that we could ask with this FTICR data. And then independently, these teams of people told 6 separate stories, and that is available in Frontiers in Water as a collation of papers. Another way that we've been able to reuse this data, I will say that the reuse part, specifically for GROW, because it's a global effort, folks have really been embracing using it to put their findings into context across the globe. And so one example of this is Tom Stack and Alexander Probst have a project where they set up stream microcosms with different stressors. And so things like different water velocity and salinity and temperature. And so they were able to ask questions in their microcosms and then see how that relates back to a more broad sense in terms of the global data. And so this is one way that it's been able to be reused. And I just wanted to highlight, you know, we're always willing to work with people to, because there is a lot more data than we're putting out. Just it's a little slow, but we're working on making that more available as we go through it. And so do reach out if you have questions on like what data is available versus what's not, because we have a person that is consistently uploading more and more data. So we'd love to work with you too. If it's not there, ask me about it. I'm happy to work with you. And then with that, I would like to thank everyone on this slide, particularly Kelly Wrighton at Colorado State University, James Stegan at Pacific Northwest National Lab, and then Alicia Wood Charlson at KBase. They've been instrumental in making this project not only happen, but also publicly available, which I'm super grateful for. Thanks.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.